Okay, so for this next section, again, I promise no slides, but that does mean you need to talk a little bit more. Though we've had a good number of questions, but this I want to be much more interactive. This section is basically about choices. Okay, you have your biological problem of interest. You've joined a lab. Your lab studies something, and you've decided I want to screen this something, and now you actually want to start planning to do a screen. What are the upstream choices that you should be thinking about as you go through that process? So we've already talked about one, so I'll start with that. So you can do an arrayed screen, or you can do a pooled screen. And I know there are some people in this room that have actually done screens, so speak up. What are the pros and cons? What are the costs and benefits of doing an arrayed screen versus a pooled screen? Why might you choose one or the other? Even if you haven't done one, what might you think would be a benefit to doing one or the other? Cost, absolutely. Cost is a main driver in a lot of projects. For a certain bucket of money, how much screening can I do? Arrayed gets more expensive more quickly than pooled in general. Each plate of virus that you make, each plate of cells that you screen gets to be, adds incrementally to the cost. Pooled virus, though, it doesn't matter too much how big your pool is. That tends not to be the cost driver of your experiment. Now, that's not to say that pooled screens are wildly cheaper than arrayed screens. It's generally not the case. But certainly if your comparison was, how much would it cost to do a genome-wide screen pooled versus a genome-wide screen arrayed, you do a lot better doing it pooled. A pooled genome-wide screen, given, again, totally dependent on how many replicates you do, what cells you're working with, blah, blah, blah. But a standard pooled genome-wide screen for a phenotype of interest, ballpark $25,000. Well, plus minus, which is not nothing, obviously. But at the same time, if you think about it, if you're doing a screen, and this is something we're going to talk a lot more about after lunch, but you need to commit to a screen. A screen is not something you just sort of do one day a week, just sort of work away at it for a little bit. Like when you're doing the screen, that's pretty much all you should be doing for that stretch of your lab life. It's not something that just sort of can trickle on in the background, like, oh, I know, I'll start in year 1 and I'll finish by year 5 of my PhD. No, you gotta work on it, very focused. So cost is certainly one. Pooled screens tend to be less expensive, especially if you want to do genome-wide. What else? What's that? Deconvolution, doing the data analysis. That's right. So pools are more complex in the sense that it's very easy to think about doing an analysis of, okay, there's one perturbation per one well. I'm going to translate that into a number somehow, eventually, but based on what you're doing. But you get a number, and okay, this perturbation caused this number. When you're doing a pooled screen, the way we do it is we do next-gen, we do Illumina sequencing to figure out the abundance of hairpins at the beginning, the abundance of hairpins at the end, and then say, okay, this hairpin increased in abundance or decreased in abundance. Okay, that's not as direct a measurement as the one perturbation, one well. In fact, the standard follow-up to any pooled screen is almost always, okay, now I'm going to do an arrayed screen on a much smaller set. So you screen the genome in a pool, and then you say, okay, here are my hits, and now I'm going to look at them in arrayed format. So there's an inherent, I understand it better if you're looking at it in arrayed format. So you want to eventually get to that. What else, pooled versus arrayed? What about types of questions you can ask? What's the difference?
Do you need to have a different readout? You can't have a cell death or live dead.
Exactly. So Monica mentioned readout. What are some readouts that you can do in a pool or an array that you can't do vice versa? So live dead you can actually do both in pooled and arrayed. So in arrayed format, some of the standard assays are a cell titer glow, which is an ATP-dependent assay, an Alamar blue assay, which is resizurin. It's this dye that gets processed by the mitochondria or gets reduced by the mitochondria. So you can certainly do live dead in an arrayed format, but you can also do live dead in a pooled format. You look for, and this is what Project Achilles is. It's the idea that, okay, we take a pool of hairpins, put them onto cells, we know what hairpins were there at an early time point, and we ask what hairpins are depleted at a late time point. So you can certainly look for loss of signal in a pooled screen as well. So you can do live dead that way. What are some things you can only do in an arrayed format where a pool just wouldn't work for you? What are some types of assays? Selection readouts, yeah, so the exact reporter you use. There are some reporters that work very well in pooled screening. So we've done many screens, and you'll see examples of this, where FACS is your readout. You use FACS to separate the winners from the losers. You start with a population of cells that's GFP negative. You're interested in the few that, for whatever reason, now become GFP positive, and you suck those guys out, and you see what hairpins are in that pool. But let's say your readout is something more sensitive, like a luciferase assay. You can't do luciferase assay in a pool. There you have to do in an arrayed format because there's no pooled, there's no light-based FACS sorter. Well, what are some other things you can only do in arrayed? So some reporters, some phenotypic stuff, right? So certainly, what's that? Imaging, yeah, imaging is a great example of things that can only work in an arrayed format. Because in a pooled format, yeah, you can certainly infect your cells and take pictures of what they look like, but you have no way of knowing, oh, this cell has what hairpin in it, right? You can only do that where all the different perturbations are separated by well. So imaging screens basically have to be done in arrayed space for sure. Anything else? Yeah. Yeah, so is there anything that the pool is preferable for other than cost? Absolutely. Let's say you wanted to do an in vivo screen, okay, where you're taking hairpins and you want to know what do these hairpins do to a mouse. Okay, now you could have one hairpin, one mouse, but that would get pretty expensive pretty quickly. You can, however, take a pool of hairpins, put them into a mouse, and see what happens. Do we do that? Yes, we do that. Yes. Wow. Awesome. Yeah, so there are a lot of, in a mouse, you can't really conceive of doing a genome-wide pooled screen in a mouse because of numbers. And we're going to talk far more about numbers in a little bit, the specifics of not putting a population of hairpins through a bottleneck in your screening. So you wouldn't do a genome-wide screen in a mouse. But we've certainly done many screens where people take a candidate set of hairpins and do some mouse model with them and see, let the winners and losers sort itself out. So obviously a lot of this is done with xenograft type assays. You take a collection of cells, human cells, put them into a mouse and see which hairpins promote metastasis or cause the tumor to regress or something like that. But yeah, in vivo experiments can be done with a pool, but can't really, cost effectively at least, done in an arrayed format, one mouse per hairpin. Dave, did you want to add something?
Yeah, so one thing you can do in pools that you can't do in plates is anything that requires tricky passaging or culture conditions. So in an arrayed screen, you're confined to whatever you can do in microtiter plates. So you don't want to be passaging the cells. Maybe with suspension cells you can get away with a little bit of that. But it's conventional cell culture in a pooled screen. So anything you could do in traditional cell culture is tractable as far as how you handle your cells. You can keep them going for weeks if you wish to, if that's what you need to model the biology that you're trying to study.
Yeah. Yeah.
Now we discover a new effect is there is competition because with pool screen, say you have 100 virus and produce 100 different effects, then there is competition of these things. Say you're throwing a parasite or pathogen.
Yeah, so the point is in a pooled format, the hairpins are obviously all competing with each other. Yeah. So you can certainly, we've seen screens where that can work against you in the sense that if you have a few hairpins that are really potent at the phenotype, those are the only hairpins you can really study because they quickly overwhelm the pool. And what related to that, what if what you're studying, there's some cell non-autonomous effects, right? I mean, let's say that you're studying something where a cell secretes something, and that secreted factor can now affect all the cells around it. In an arrayed screen, no big deal, because all the wells and all the cells in that well got treated with the same perturbation. But in a pool, now one cell could be affecting all of its neighbors in a way that could really confound your analysis. So if you're working on cell non-autonomous effects, that can also be a big confounder. Another choice I wrote up there, positive versus negative selection. So what does positive versus negative selection mean? When there's some screens, like if you're doing an imaging assay, the idea of positive versus negative selection doesn't really come into play. But But what does positive versus negative selection really mean? Can we give a good example of that? Let's say you're thinking specifically about a drug of interest, like some chemotherapeutic. What would positive versus negative mean? Related to survival, yeah. It doesn't have to be related to survival, but it often is. So a standard experiment can be to take a drug of interest and to say, okay, I have cells that are sensitive to this drug. Okay, so if I add this drug to the cells, they all die. And now I'm looking for a perturbation that allows them to survive. So that's a positive selection that you're gaining a phenotype, but from the screening standpoint, you're gaining signal, right? If you add your drug to a well of cells, in every well they should die, so you have low viability signal. But then the guy that's a hit is an increase in signal. Okay? So that'd be with looking for resistance to a drug, the perturbations that allow resistance. The converse of that is sensitivity. You start with a dose of drug or a drug where the cells are resistant. So when you add the drug, all the cells grow, and you're looking for the few perturbations that now allow the cells to die. So that's a loss of signal, a negative selection type of screen. Those in general are harder to execute. That doesn't mean you can't do it. I mean, all of Achilles is a negative selection screen. You're looking for the guys that drop out over time. And it can work great, but just practically speaking, the positive selection screens, because you're looking for a gain of signal or a gain of a phenotype, which don't always correlate, but gain of signal, they often are easier to execute, and you have more margin of error usually in setting up your assay. So that's one difference, a gain or a loss of signal.
But
Yeah.
Are they related to pooled or array, or is it equally difficult in one or the other?
Yeah, so is that related to pooled or array, or is it equally difficult? I'd say it doesn't necessarily relate to pooled or arrayed. I mean, I'll put it this way, you can definitely do, and we've done, both positive and negative selection screens in both arrayed and pooled format. Again, they're usually the positive selection screens are easier to execute, meaning you have more room for error. But we've certainly done both. One thing, just while we're on the, go ahead, Dave.
Yeah, I'll jump in on that positive and negative selection. I think it might be useful to think about why positive selection might work better. And I think that it does apply to both pooled and arrayed circumstances. I think it's all about how uniform your non-hit cells are. Okay? It's easier to see hits if none of your non-hit cells look artificially like hits, false positives. And they're just, in most technical situations, it's less likely to spontaneously acquire a signal for a background cell than it is to, there's more ways that a cell could fail to produce the signal by accident. You could get a false loss of a signal. Then there are ways to get a false, a positive signal. That's not always true, but it's often true, and I think that's why positive selection, it typically works better.
Yeah, and related to that, especially since a lot of people are interested in taking a drug specifically and learning more about its mechanism of action, or learning, in the context of oncology, okay, we know this drug works, but what combinations might it work better with? I mean, that's a pretty standard question. Picking the right dose of drug, I mean, this is more of an assay development thing, but a general rule of thumb is that it's really hard to get a screen to report in both the positive and negative direction. So it's very easy to think about in terms of a drug dose curve, right? I mean, if you think, okay, you have a drug of interest, okay, and here's its dose titration, something like this, okay? So this is increasing concentration of drug, and here's something like cell survival. Okay, at a certain dose, here's your IC90, here's your IC10, here's your IC50. No, excuse me, this is IC10, 50, IC90. Okay, so at this point, you're killing 90% of the cells, this point 50, this point 10. If you attempt to thread the needle and say, aha, I'm going to try to work at the IC50 and try to find things that work in both directions, you will fail equally well in both directions. It is far better to say, okay, I'm going to look up here for things that shift down here or vice versa. We've very rarely seen, not like it never happens, but it's usually pretty rare for a screen to report to be equally powered in both directions. So it's usually worth thinking about, if you really are interested in both directions, then do 2 screens, one to look for one to look for the other. But it can often be quite hard to, in a single screen, go both ways. Yeah, so drug selection, so what are some other choices? Well, yeah, as I mentioned, yeah, Monica.
How often, or are you doing a lot of these modifier screens where you use a small dose of the compound and then just see which of the hairpins to push it over? Is that something, I know of one, but is that something that's become more routine or people do more of?
Yeah, so the question is, how often are we doing these sorts of modifier screens? And there's certainly been in the BroadE community examples of them working quite well, and it's a question of, are we doing more of them? Now, that somewhat relates to how we work as a platform in the sense that we think they're good screens, and we now have experience in how to execute them, but someone else needs to say, okay, here's the drug I care about, here's the cell I care about. Yeah.
Are you seeing a lot of people that care about these types of things, or do you really see it just internal because we talk about it and think about it all the time, and we push people to, think about this type of screen, but is it something that people have started to think more about, or they want to do, or is it just when we push them because we've heard of it?
I think we're seeing an increase in the number of modifier screens that are being done over the last couple of years. I don't know whether that's because we're promoting that they can work very well and encouraging people, or just more people are showing up with that in mind already. But we are doing more of them.
Is fairly decent.
Yeah, it's pretty good.
Yeah.
I mean, the really bad case is when you fail late in the game. There's a lot of these ideas floated that might not work, but you figure it out early on, and that's a fine thing. But the ones that we really pursue and chase very hard, the success rate's pretty good.
Yeah, and again, a lot of it is setting up your assay right so that even if it does fail, you at least know it failed, or that okay, the biology I was studying, there are no perturbations that do this. So you being able to believe the fails is an important. It's an important part of any experiment, but especially a screen, because it's such a time investment. Yeah.
Between, for example, Achilles and the pharmacologic screens be done the formal, because I always get this question, when you do like those synthetic, quote unquote, right, synthetic screens. If you think that your effects are on target, now we have a lot of pharmacologic data from CTD, square and so forth, like to say, okay, this is what overlapping with those,
Yeah. So the not really a question, just a good point that we do now have in this small molecule hairpin space just lots of data sets being generated, and integrating them is certainly, it sounds great in theory, and it will be great once we can actually do it. But the integration of data, everyone here who's been at the BroadE longer than a year is just smiling and nodding right now. Integration of data and getting it out of silos and getting it to talk to each other, that is a severe software engineering problem, and it's something that we're all aware of. It doesn't mean that we've solved it yet, but it is, because exactly, that's exactly what you want to be able to do, push button wise, but it's not fully there yet. But it is something that, because that's certainly a good point. Paquita. Yeah. Yeah. Yes, it's that's the dog group. Yeah.
Yeah, I think that it's fair to say there are a number of efforts to try to integrate various types of data, and certainly some of them have already succeeded to various degrees. It's just there's not a good general way to make all data sets easily interchangeable for a biologist. If you try and build a lot of versatility into the system, it gets too hard to create. But certainly there are lots of success stories of people trying to integrate 2 types of data in a very specific way, and they pull that off just fine.
Yeah. But it's absolutely a challenge, absolutely. So choices, getting back to this. No, this is totally fine. Choices, so we talked about how we have an shRNA library and we have an ORF library, and at least for a while they were incompatible in the sense that if you want to do a pooled screen, you could only do it with shRNAs. If you want to do an arrayed screen, you could still choose ORFs or hairpins. When might you choose one versus the other? And I'll add the subtext of, is that kind of a false choice? Do you have any strong opinions on when you would do one versus the other, or why not just do both? Or if you only have enough money, which one should you do first? You know, what do you think? That's what you wanted me to tell you, yeah. So, as I alluded to, I do think that is a bit of a false choice. I mean, I think that there are some examples where if your model is, let's go back to that 5q example. If your model is, I want to know which gene is implicated in loss of the same phenotype as loss of part of a chromosome, there's not a lot of reason to think that overexpressing something would mimic that phenotype. That's where you think, okay, I want to use a small hairpin library to see something go away. So that's a clear example. Or vice versa, let's say you had some chromosomal amplification in some cancer phenotype. Okay, I want to overexpress ORFs and see which one mimics that. But I'm interested in how the mitochondria functions, or I'm interested in neuronal guidance. There's all sorts of phenotypes where gain or loss, there's no obvious reason to think that you would only find hits in one but not in the other. I think people are afraid of that. Although, as we'll talk about, usually the problem is too many hits, not enough hits. So I think that is a bit of a false choice. For all things being equal, if you could do both a pooled screen in shRNA space and ORF space, absolutely do both. It's not twice the work, for sure. You're doing assay optimization the exact same way, whether you're doing shRNAs or ORFs. So there's no reason not to do both. So I think it's a choice that, the reason I bring it up is that I think it's a choice that Dave and I have talked to a lot of people about, because people worry about making the wrong choice. But I think there are very few examples where you could make a wrong choice, or that you could think a priori, okay, is it going to be hairpins or ORFs are going to be better? And I don't think that's really panned out.
Yeah, I'd be very suspicious of people's guesses that you're going to see a stronger phenotype from loss of function or gain of function. I think it could be pretty hard to predict that. There might be, if you have to pick one or the other as your primary screen, say for cost reasons, you're quite likely to do the other direction as a follow-up on the same genes, and that's a good idea. But if you have to pick one or the other, there may be some practical things that guide you one way or another. I would say if you have a positive control based on overexpression of something, you might want to set up that screen because you have a positive control for that particular screen. If you have a positive control that's a loss of function phenotype, you might want to do the RNAi screen, the loss of function screen, and take advantage of that. So there's some practical reasons that might push you one way or another, but a priori knowledge that these must be, I can only get this activity in this complex process by adding protein versus by taking it away, or that's hard to tell usually.
Is there an economy of scales in doing both?
Is there
and you see a lot of people doing both simultaneously.
Yeah, so is there an economy of scale in doing both? I can't answer yet in pooled space because, the pooled ORF library is only coming online soon. It's not ready yet. Although certainly anyone who screens with me, I will encourage them to do both because it really is the exact same assay development. In a RAID space, it won't be exactly the same. And I don't know of nothing's coming to mind where someone did in parallel, like first I'm going to do my shRNA screen, then I'm going to do my ORF screen, like weeks 1, 2, 3, and then 4, 5, 6. I don't know of any examples of that.
No, two different postdocs in the same lab,
That's
for
true,
example.
yeah.
So the economy, I think, comes from the fact that some of the assay development is going to be, could likely be similar. Yeah. And so you might not have to start over from scratch for the second version.
And you get a big bump in the ease of follow-up when you have both combined. So the hardest thing in all of these things is always the follow-up, right?
That's right.
So do you get a big bump in how easy or how clear the road downstream is when you do both?
Yeah, so do you get a big bump in follow-up if you've done both the shRNA and the ORF? And I think I'll have to answer now that theoretically yes, but we just don't have the data to say so. But certainly, I mean, look, if you were to do a screen and saw, okay, the knockdown, I see one phenotype, the ORF, I see the opposite phenotype, like, you're golden, right? I mean, that's a gene you're really going to hang your hat on. But we just don't know yet how often that occurs, but we're very excited to find out.
Yeah, I mean, in a lot of cases, you can do the other screen as a follow-up on the hits from the first one, and then you will have the data in both directions for that set of genes. The thing that you lose by not doing them both as large primary screens is the cases where maybe you have a decent but not very top of the list type hit in both screens. You might pick that if you know about it up front, but it'll never make the hit list. So it's hard, that you can't know ahead of time, but you can always do both on a selected set of follow-up genes. Yeah.
Yeah.
And the consideration of the positive and negative signal, and like there's no screen that can go both ways. If you do both ORF and shRNA, ideally you would actually, what makes reasonable sense is that both screens will give you entirely two different lists of genes.
Right. So Xiaoyun's point is, related back to what we were talking about with positive and negative selection. The ORFs are likely to work in one direction, the shRNAs in the other, meaning you're overexpressing versus knocking down for sure. But if you did the same screen, like let's say the drug selection example, if you're looking to go from IC90 to IC10, would it make sense to do both shRNAs and ORFs, or should you do?
I think it makes sense, but it's not doing both for a sense of, cross-referencing each other.
Right. No, that's exactly right. I mean, so if you were to do, let's say, that IC90 to IC10 example, you would not expect to see the same ORF scoring as knockdowns, because that would be hard to interpret, right? I mean, there you'd have to invoke some dominant negative phenotype or something. So you wouldn't expect the same perturbations or the same genes to score, both moving in the same direction, knockdown or overexpression. That's exactly right. So you would need to do, if that's what you were looking for, you need to do two screens. One, the IC10, one 10 to 90 and one 90 to 10, and see the opposite phenotype. But I think in that case, the choice is, what is more likely to cause the 10 to 90 change in ORF or in shRNA? And I think that's, again, as I've said, I think that's hard to know in advance. And I think there's no reason to think that an ORF would be better at it or worse. I mean, certainly with the case of vemurafenib, a BRAF inhibitor, where we did do an ORF screen and an shRNA screen, and there were shRNA hits and there were ORF hits that showed the same phenotype. Now, they weren't the same gene, but that makes sense. Kina?
Yeah, I was going to add to Shelby. I think she's right, but at the same time, you could also do a pathway analysis, and you imagine that maybe that will come up.
That's right. So the idea of doing pathway analysis and realize, okay, it rarely works like this, but okay, you find that the knockdown of the kinase shows you one effect, but the overexpression of the phosphatase that affects that kinase shows the effect as well. Absolutely.
Well, not just be, when you do those screens, you usually do it in one cell, because they're big scale and expensive and so forth. So you have to be a little bit careful about the cellular context. Like if you were to do it in the same, because the example you mentioned for cancer, like if it's a natively resistant cell line, in which case you would be looking for synthetically healthy with knockdown. That's not going to be the same cell line as something that is like very sensitive and you're looking for resistance.
Yes,
exactly.
exactly.
The screen starts with two different cell lines. So you have to be a little bit careful. If it's lineage specific, maybe.
Sure. So let's talk about this specifically. So you brought up the point of cell lines, and this somewhat relates to large or small scale, but let's change it to the idea of depth. How wide do you go, right? I mean, we do know context matters incredibly. If you think that, I'm going to screen HeLa cells and I'm going to learn about all of cancer. No, you're going to learn about all of HeLa cells. This is something that Project Achilles has taught us very well, but many other screens too. If you switch cell lines, it's a whole different world. What you can't do is say, okay, I have one cell line that has a KRAS mutation, one cell line that doesn't. I'm going to do a screen in both of them and think that you're learning something about KRAS mutational status. Because the difference between cell line 1 and cell line 2, yeah, it's KRAS, but it's also 5,000 other things that you don't know about. So certainly, cellular context matters a lot. So that's a big question when setting up a screen. How wide do you go on cellular context in the primary screening? Do you do one cell line that you think is a great model for what you're studying? For some things, yeah, maybe. I mean, let's say you're studying metabolism, okay? You're really interested in Krebs cycle or something. You think, okay, or translation, something that's likely to be quite similar across a bunch of different cell lines. There, the cell line you pick up front might not matter too much. But there are going to be other phenotypes that are incredibly context dependent. Basically anything having to do with cancer is going to be extremely context dependent. So thinking up front, okay, if I do this screen, should I do my primary screen in one cell line and then do follow-up in 8 different cell lines? Should I do the primary screen in 8 different cell lines? So where you make that cut of how broad do you go up front is a major variable.
And just to make sure, the choice that John framed there there's something implicit in there that's very important. It's not a choice of whether you test your biology in lots of different model systems. It's what order you do it in. Yeah. So you will inevitably need to test to make sure that the findings that you're making are robust, aren't HeLa biology or 293T biology. You want it to be relevant to mammalian biology. But the order that you do these things in could have a big effect on your productivity, on how much it costs and the yield of your screen.
So I said we'd go for about half an hour, and then I promised another break before we pick up with the next section, which is more PowerPoint slides. So why don't we take a 15-minute break here. We're going to come back. I mean, this whole next section is all about the how to do screens, how to interpret them, how to follow up. So we're going to, all of these things are going to come back up multiple times in the afternoon session. So if you have more questions or comments or think, there's an idea there, but I didn't get all of it, hopefully in the afternoon you will start to get all of it for these various choices and decisions one makes in setting up a screen. So let's take